Okay, well, thank you. I want to thank the sponsors for inviting me to come speak with all of you. So just a little bit about my personal evolution. So about 20 years ago, I started as a geneticist, and then after about 15 years ago, then I became like a genetic molecular biologist, and then about 10 years ago, I became a genetic molecular biologist toxicologist. So I kind of have just like moved, I kind of just like added like a Legos, is like added one piece on a time. And it's kind of like similar to what I see, a story I see very similar from a lot of you is I talked to a lot of people that were in like traditional medicine, and they did that for maybe like 10 to 20 years, and then they like figured out that they're like either their own personal cases or cases from their clients that they're like dealing with, that they couldn't solve everything using traditional medicine. They saw like there's pieces of puzzles missing. So one thing that through my work and the work of my company is that we are trying to like help you find those pieces of the puzzle of how can we give a personalized medical program for every single person. So just like one piece of information. So yes, I'm the associate lab director at the Great Plains Laboratory. So I mean, I do have a little bit of a bias here, but I'm going to try to give as broad spectrum talk of how you can like add different testing to your panels for your patients to give a personalized care. And today we're going to mostly be focusing on genetic testing and the organic acid test. The organic acid test is kind of like the hub of a wheel, where it kind of like everything like centers around it, where you have like genetic testing, you have detox testing, you have different type of food sensitivity, all going around the organic acid test. To get a really good personalized care package, I really say that there has to be like three things in the center of it. And those three things are talking to your clients, the organic acid test, and genetics testing. And those three things really have to like work in concert to get like a good like personalized care for the patient. So today we're going to talk a little bit of background, and the background is going to be mostly focused on what is organic acid testing and what is next generation sequencing. Then we'll talk about how genetic testing is utilized. Like most of the time you're going to hear a lot, and I'm sure that you've heard a lot in the last couple of days of SNPs, which are single nucleotide polymorphisms. Then we'll talk about how these SNPs overlap with the organic acid test and how they affect like methylation, neurotransmitter metabolism, detoxification, oxalate metabolism. So the organic acid test is focused on, this is what, it's 75 different markers here. And the first page here is kind of focused on gut health, which is mostly on yeast and fungal markers, bacterial markers, and Clostridia markers here. The first top part here is mostly on fungal markers, mostly Aspergillus. So if you have elevations in these markers here, it could show that your patient has like overgrowths and different like fungus. And a lot of times this can be caused by exposure to different molds, such as Aspergillus, Penicillium, Fumaricin. A lot of different mold toxins can show like positives in this section. Then you have yeast here in markers 6 through 9. Then you have bacterial markers in 10 through 18. The next section of the organic acid test centers mostly on oxalates. Then you do Krebs cycle and mitochondrial markers, and these are really important for trying to assess whether or not your patient has environmental toxicity. And we'll talk more about which markers you really want to look at there to try to decide if you need to do more metabolic toxin testing. Then we'll talk about, the next section is neurotransmitters, ketone and fatty acid oxidation, vitamins, and detoxification markers. And we'll talk about several of these today because a lot of them focus on genetic testing and methylation. So how does organic acid test work? So really this is a metabolomics type test where we run a lot of different molecules through a gradient here, which is a copper tube. It's about 15 meters long, and all the compounds come out over a 10-minute period. And every time a compound comes out, you can see abundance, which is different, like different heights of peaks here. And each one of these peaks here is a different compound. You can focus in on one particular peak here to get this pattern. And this pattern here shows different molecules and different masses. You can look at these combination of different masses, and they'll give you like kind of like a fingerprint of what molecule is coming off the column at any one particular moment. So this will give us like 100% accuracy of what molecule is coming on. So the intensity of the peak gives us the abundance, and then this pattern gives us the identification of what the compound is. The next part of the thing that we wanted to talk about was next generation sequencing. There's really two different forms of sequencing out there. You kind of have a TaqMan-like sequencing, which uses fluorophores, or you have the new type of technology, which has been kind of like coming around in the last like 8 years, and this is called next generation sequencing. The fluorophore type of sequencing uses probes, and it'll give you like a color change in a plate of whether or not you have different molecules. Like say, if you had a G, it'd give you like an orange color. If you had an A, it'd be green. If you had a T, it'd be red. It'd give you different colors depending on which nucleotide you had at a specific spot. But for the next generation sequencing, it actually sequences that stretch of DNA about between 50 and 100 times, and it gives you about between 99.99% accuracy and 10 to the ninth amount of accuracy. So you have a lot greater accuracy using the next generation sequencing than TaqMan. Most of the sequencing that we've like used in the past at Great Plains have been the next generation sequencing type of technology. So when you sequence something, you are really sequencing a gene. So just to help you all understand what a gene really is, a gene has multiple different parts to it, kind of like a street, where you have this is the kind of like where the start of the street is, and then you have the stop over here on the right-hand side. Here's where your red light, green light section is. This is the regulatory section. This allows the body to know, does this gene need to get transcribed in this type of cell? Because not every gene is transcribed in every type of cell. Obviously, eye cells make different type of proteins than liver cells do. So you have to like look at this and try to like coordinate. Then after this regulatory region, you actually have the coding region here. This tells the body what protein needs to get made. The exon parts are taken out of the gene, and the introns, the exons are the ones that are coded for, and the introns are the ones that are removed. So for humans, we're only looking at the exon part of the gene. And that leads us to SNPs. So SNPs are single nucleotide polymorphisms, and there's different types of SNPs. We're going to look at a couple of these different examples here. So like for a normal spot in the genome, you could have about 95% of the population having an A at that particular part of the genome. Let's say like 4% of the population has a G, and 1% of the population has a T. This is very common. Now, sometimes you'll have like a 50-50% split where one half the population has like an A and the other 50% have a G. Depends on how like conserved that particular part of the genome is. These changes in the genome can either have no effect or really big effect, and we'll talk about why that is. The no effect can be caused by something called a silent mutation. So all the complete genome here looks at different like base pairs, and like all the base pairs are like A, G, T, and C, and these base pairs code for different amino acids. So like ACA codes for the amino acid cysteine, but there's some redundancy in the code. So like ACG also codes for cysteine. However, you can have something called a missense mutation, where you have like ACA codes for cysteine, but then you could like change this A to a G. Now it's GCA. This codes for a different amino acid called arginine. And these missense mutations can either like have like very little effect, because some amino acids are very similar. Like some of them are hydrophobic, some are hydrophilic, some are very large, some are very small. So you could have two amino acids that are very similar in structure and properties, but then you could have two amino acids that are very different. And the ones that are different can cause very different changes in the protein and its structure and function. The worst type of mutation that you can get is something called a nonsense mutation. And this also depends on how, like, where it is in the genome. So nonsense mutation goes to, like, if you have like an ACA, which codes for cysteine. If you change this A to a T here, it codes for a stop codon. The stop codon is kind of like a stop sign. And you really want the stop sign to be at the end of the protein, or at the end of the gene. But sometimes if you get these mutations in the middle of the gene, it can cause you to have a non-functional protein, because you're only making half the protein. Usually a protein that's only half the size is really non-functional. So that leads us to the first section here that we were going to talk about, methylation. There's a lot of different genes that are really important for this cycle, and the reason why this is it's very highly regulated. So you kind of pathways that are very highly regulated usually have a lot of different enzymes involved with them. We're going to talk about a lot of these today, such as the MTHFR, the MTR, the CBS, the SUOX, the AECY. We'll talk about like different like ways that you can counteract polymorphisms in these genes and what the polymorphisms can lead to. So again, these are kind of just to show it in more bullet point form here. Is you get the MTHFR, MTR, SUOX, MTRR, BHMT, CBS, and AECY. And we'll lead with MTHFR first. And the reason why this is because it's the one that's mutated the most often. You're going to see a lot of patients with MTHFR polymorphisms. And these polymorphisms can cause a lot of changes in the activity of the enzyme. This table here is really indicating how prevalent these mutations are. What you're seeing here is like a majority of patients probably are in the Caucasian and maybe in the Asian groups here. And you can see that only about half of them are in the wild type homozygous, meaning that they have two of the active versions of that enzyme. If you have a patient with the C677T that they're heterozygous here, that's like about the other half of the 50%. And then the patients that are homozygous variant can range anywhere between 4 1/4 of the total population. And then for Asian, it's about a fifth. And these mutations can cause a lot of loss of activity here. Like we were talking about the C77T here. So the wild type has 100% activity. But if you have one variant copy, you lose about a third of all the activity of the enzyme. If you have two variant copies, you lose about 3/4 of the activity. So that's a lot of activity lost. If you have the 1298 heterozygous, you only lose about 17% of activity. But then if you're a double variant, you lose about 40% activity. If you're double heterozygous, meaning you have one variant copy for both of these different alleles, then you're lost about half of the activity. I have a lot of patients that I deal with for when I do consults that have their homozygous variant, and they can have other heterozygous for the other one. So that's kind of like falling into this group here. And those patients can really have a tremendous loss of activity of this enzyme. In the type of like symptoms that we're seeing here are like patients that deal with a lot of infections, a lot of inflammation. They can have trouble with like cancer, lots of brain fog, decreased amount of energy, lack of B12. So, I mean, you can have a lot of different patients, and a lot of them like manifest differently. So it's not just like every single patient suffers the same type of symptoms, because a lot of it, they have different environments. So this is where you're kind of like looking at like at a multiplication equation where you have the genetics multiplied by the environment equals the outcome. So this is what you're really like looking at here. And the reason why a lot of your patients have different symptoms depending on what their environment is. Obviously, a lot of you have like dealt with a lot of patients with methylation issues, and you know a lot of the different symptoms like schizophrenia, heart disease. Part of the organic acid test that there's several different markers that I look at for trying to like see if whether or not a patient is suffering from a decreased amount of methylation. One of the ones I find that works the best is this marker 59 here, the 2-hydroxybutyric. The uracil marker can also be used, and I'll see patients that are elevated for the uracil marker, which can also be an indicator of methylation deficiency. There's two things for the patients that are homozygous variant for the MTHFR. There's two things I really recommend them starting off with, and this is starting off with the methylcobalamin and measuring the homocysteine, which will give us a better like assessment of whether or not the patient is really needs some more methylation help. One caveat to measuring homocysteine, however, is that if your patient is experiencing a lot of environmental toxicity, this homocysteine measure could be artificially low. And the reason why that is because homocysteine, which we'll talk about a little bit later, gets converted to cysteine, which gets converted into glutathione. If you have a patient with a lot of environmental toxicity, you could be like funneling a lot of the homocysteine down that pathway, and that could indicate that the homocysteine might not be normally being regulated. And so you could get a false like negative or false assessment of what the methylation state is for the patient. So the next enzymes that we're going to talk about here are kind of the combination, because they kind of like work on the same part of the pathway here, is the MTR and the MTRR. And one of the like common like SNPs that are looked at in a lot of genetics tests, and this is one that we like offer, is you're looking at this like panel here. For like MTR, there's really like one major SNP that you're looking at, which is this like A2756G. And then you got these like 6 other MTRR SNPs. They're different degree of loss of sensitivity or activity for these particular SNPs. There's a lot of different diseases that are associated with these SNP polymorphisms. It's probably some of the most. So we talked about the C677T from MTHFR and the 1298. And then for the MTR, you got this 2756. And then the most common and most problematic for the MTRR is this A66G. And a lot of these can cause the risk of folate deficiency. And like I said before, patients that have polymorphisms in either the MTHFR, the MTR, or the MTRR, a lot of these can like result in the accumulation of homocysteine, which you can see for this particular patient here. This is a very like limited and obviously not completely inclusive symptoms that you can see for these polymorphisms, such as like leukemia, Crohn's disease, heart defects. And the reason why we're going to talk about leukemia more is like in a lot of other cancers is because if you have these lack of methylation, it causes depletion in the amount of glutathione. So we'll talk more about that later when we hit the detoxification part of the organic acid test. Again, other things to like really like be like cognizant of when you are measuring things on this test, and what things to avoid is so you want the patients to take the methyl B12. You want to avoid certain things too, such as antacids, potassium chloride, gout medications, antibiotics, metformin. All these are different like medications that can cause B12 to become depleted. The next section that we're going to talk about today is BHMT. And so where this one fits in is that you get homocysteine. Homocysteine can go in like lots of different like directions here. We like talked about towards cysteine and glutathione. It can also go towards methionine. And the methionine here is the BHMT. And the cofactor for this BHMT is betaine. It's also called TMG. There's several different polymorphisms that can cause disease here for BHMT, and you can really see homocysteine levels becoming elevated for patients that have polymorphism in it. The reason why this is here is because if you're looking right here, the homocysteine needs to get converted to methionine. If this polymorphism is canceling out this reaction, then you're going to get it's going to cause more of it to stay as homocysteine. There's 4 different polymorphisms that you really need to be aware of, and all of them cause significant decrease of activity of the BHMT enzyme. Some of the symptoms that you can see for patients with these are like ADHD, obesity, fatty liver, diabetes, hepatocellular carcinomas. And so as we talked about earlier, the cofactor for BHMT was the TMG, what's also called betaine. You can also, other cofactor is zinc. These 2 items can help increase the amount of activity for the BHMT enzyme. One of the possibly one of the most important enzymes we're going to talk about today is the CBS enzyme. As you can see it here, it's really it's causing the homocysteine to be converted over to cystathionine, which then later gets converted to cysteine, and then eventually got down to glutathione. And glutathione possibly is one of the it's definitely one of the most important molecules for human health, at least for detoxification. And you can see here, we're looking at patients with polymorphisms in the CBS gene. So here is wild type individuals here over on the left, and here is the patients with the polymorphism of CBS. And you can see here a decrease what the CBS activity is. And the people with the CBS polymorphism have a lot less activity in the CBS enzyme. This can result in a decreased amount of glutathione here. So you can see here, this patients that have their wild type have 7 nanomoles per milligram of protein of glutathione. You see about 30% decrease of glutathione in the patients with the CBS polymorphism. This can become even more acerbated if you have patients with extreme amount of exposure to different toxins, such as like mold toxins, rubber toxins that bind up glutathione, plastics bind up to glutathione, metals can bind up to glutathione. So exposure to any of these things can further acerbate this loss of glutathione produced by the CBS polymorphism. So how do we help treat patients with CBS polymorphisms? Really, the things that we've seen to help the best is trying to support this enzyme. This can deal with supporting the B12 and especially the B6. I see a lot of patients that have the CBS polymorphism, and what I'll see very frequently here is if you have a patient, this is a particular patient that we did genetics test on, you can see here the polymorphism right here, the A360A, and what I see for a lot of patients with the CBS polymorphism is they really have no B6 at all. And the reason why this is that the CBS enzyme is trying to work extra hard and it uses excess amounts of B6. So most patients are using up a lot of B6, but these patients with the CBS polymorphisms use B6 up even at a greater frequency. So you really need to support them with the B6. And so the first thing I really try to do is do this 50 milligrams of B6 per day. I also will try to support with the 50 milligrams of B12, but the B12 is not as important, and some patients don't respond well to B12, so I don't always do B12 with every patient, but the 50 milligrams of B6 is very important for the patients with the CBS polymorphism. The next enzyme that we're going to look at here is the vitamin D receptor. The way the vitamin D receptor works is it actually forms a heterodimer with another enzyme called RXR. So VDR will hang out in the cytosol, which is outside the cell, until it binds the activated vitamin D. Then it'll transverse into the cytosol, into the nucleus, form a partnership with this RXR, and then it'll bind to these vitamin D receptor elements, which then causes a green light to be turned on for the coding for about 6,000 different genes. So you have about 6,000 different genes that are being turned on by this VDR RXR heterodimer, which binds to these VDR responsive elements. The thing that I see a lot happening with patients is that they have polymorphisms in these VDR elements, which prevents them from binding to the activated vitamin D. So we'll do a vitamin D test in the blood, and we'll see that they have plenty of vitamin D in the blood, but we'll notice that these VDR responsive elements are not being activated properly. And the reason why this is because the VDR elements can't bind to the vitamin D. And if this happens, you can see a lot of different diseases that are kind of similar to a patient that has vitamin D deficiency, such as types of Crohn's disease, things that are similar to cystic fibrosis, liver disease, obesity, renal failure, different types of cancer or autoimmune diseases, vitamin D resistant rickets. And really the way to like get around this is that we've seen is like to treat with either like 5,000 units of vitamin D per day, or possibly up to like 10,000 units of vitamin D per day. And really the way that you can tell if whether or not it's working or not is the marker 75 on the organic acid test, which is looking at phosphoric acid. Phosphoric acid is one of those compounds that are downstream from the vitamin D receptor binding to the VDR elements. If this is occurring appropriately, you're going to release phosphoric acid out of the cells, and it's going to enter in the bloodstream and then eventually into the urine. So you're going to see the marker be here somewhere around 2,000. If this isn't to happen appropriately, it's going to start getting low. I would consider this patient low. Really, I consider anything under 1,500 low on marker 75. And so for these patients, I usually recommend doing about 5,000 to 10,000 units of vitamin D. So now we're going to move a little bit away from methylation and down more into like one of the like pathways that are downstream from methylation, and that is the production and metabolism of neurotransmitters. So when we were talking more earlier about methylation, we talked about MTHFR, but then if you notice, there's like several different like endpoints for methylation. We touched on glutathione production, but other important ones are the production and metabolism of neurotransmitters, such as dopamine, norepinephrine, over here, and serotonin, up here. So now we're really going to like focus more on the like metabolism of these neurotransmitters. And the enzymes that are really important for the metabolism are COMT, MAOA, and DBH, which is dopamine beta hydroxylase. We'll touch on all three of these in just a bit. So when you're talking about COMT, you're talking about the metabolism of either like dopamine, norepinephrine, or epinephrine. And COMT has three different substrates that it utilizes. One of them is depicted here as SAM or SAMe. And then the other two are actually lithium and magnesium are the three different cofactors for the proper functionality of COMT. COMT, if you can see here, is present in a lot of different spots in the pathway. So you have dopamine up here in the upper right, and epinephrine over here in the upper left. And there's two main metabolites that are formed by the metabolism of these. So dopamine really, its metabolism leads to the production of this HVA, which is the homovanillic acid. And then epinephrine, its metabolism by COMT, MAOA, leads to production of other enzyme molecule called VMA, which is the vanillin mandelic acid. And both of these are measured in the organic acid test. You're going to see these on the second page, markers 33 and 34. So here is a result from a genetics test up here on the top. This patient had a double polymorphism, the V158M and the H62H. And as you can see here, this can show that the patient is very low on HVA and VMA. And this is like one of those spots here where I really like, it really pays a lot of attention of like talking to your patients here to try to like figure out what this low HVA and VMA mean. Because these low HVA and VMAs can really mean two different things. The first thing, which a lot of practitioners think, is that it means that they're not making enough neurotransmitters, which it could. The symptoms that usually associate with low neurotransmitters are low motivation, fatigue, depression. But then the patients with the COMT enzymes, which these low markers could just show low metabolism, but they also have really high dopamine and norepinephrine inside their synapses. These are like your patients with ADHD, also depression, a lot of anxiety, schizophrenia, OCD-like symptoms. So that's what those type of patients are the ones that actually, if you were actually looking at their synapses, they would be over here, but we're looking at urometabolites. So they're down here because of their COMT enzyme. And so there's a lot of studies that look at COMT and how it like affects people's mood. And this is just like showing here, like this is the patients that are wild type are up here, patients that have the polymorphisms are on the bottom. The problem is that patients with the polymorphisms or the variants of COMT, it just really does not, pleasant stimuli do not increase their positive affect. And this can like lead to changes in brain structure as well. You can look at different MRIs or SPECT scans and see like different changes in like, so this is a paper looking at the COMT Val 158 methionine, and children with valine homozygous attention deficit hyperactivity disorder have altered white matter in their brain. So you can see like they actually have less white matter in their brain than patients that don't have the variant. Here's another paper from 2014 talking about different variants. This one shows that patients with the COMT polymorphism have a higher possibility of developing depressive disorders. So we kind of like talked a little bit about what the cofactors are for COMT. This slide here really just touches on to it of how to increase the COMT activity. The way that is to like supply more fuel. And I mean, a lot of these things are just like trying to like stoke a fire or stoke an engine. You need to like provide more fuel to it. So what you need to do is pretty much like start with like CME, start at maybe like, and actually this might be a little bit too high. You might want to like start at like 50 milligrams per day. And then the next thing is to provide some lithium orotate, 5 milligrams. I usually start at 5 milligrams, but you could go like to 10, to 15, to 20 milligrams. I talked to a lot of practitioners that utilize 20 milligrams per day of lithium orotate. And then finally, you want to do about 400 milligrams of magnesium. These three things can really like help like stimulate the activity of the COMT enzyme. The next enzyme we're going to talk about is the MAOA. Again, you got epinephrine and dopamine up here on top, and then there's several different spots that it can affect metabolism here. So MAOA also is really important for the metabolism of serotonin. So you have serotonin here. It will eventually lead down to the production of 5-HIAA, which is a molecule that we measure on the organic acid test. So you're looking for this 5-HIAA molecule on the organic acid test. But MAOA is really important for metabolism of epinephrine, norepinephrine, dopamine, and serotonin, all four of these. In a patient that has a polymorphism, here's another patient. These are from actual results that we've seen in our laboratory, where you do the MAOA, and this one has the R27R, and this causes lower MAOA activity, which leads to like mood swings, sleep disorder, and so the polymorphisms can go to show like low amounts of HVA, VMA, or 5-HIAA. So you see here for all three of these, you have low amounts of metabolites. There's a lot of different papers here that show that it can lead to, and a lot of times you're like looking at the same type of things: ADHD-like symptoms, anxiety-like symptoms, depression. This paper's conclusion is that taken together, these results suggest that the genetic of the MAOA mutations lead to depression symptomology through brain function associated with experience of negative emotion. So, and the other thing about these patients is they can be more likely to form like drug addictions because they're trying to self-medicate. Ways to deal with these particular patients are you want to decrease their amount of amino foods, such as like fish, cheese, fruit, such as bananas and avocados, chocolate. There's ways that you can actually increase the activity of MAOA, such as like giving them progesterone. You can give riboflavin as well. Is probably the one that I do the most. Besides the dietary changes, I probably recommend riboflavin most often. Inositol also can help because it reduces the 5-HT2A activity. Other enzyme that we are very interested in at the Great Plains Laboratory is dopamine beta hydroxylase, which is right over here, which is very important for regulating the amount of dopamine versus norepinephrine. You can see the graph here where you go from dopamine to norepinephrine, and you're using actually two different cofactors. The first is ascorbic acid, vitamin C, and the second is copper. Copper is not utilized in this reaction, and copper is very highly controlled in the body, so I very infrequently see patients deficient on copper, but I see a lot of patients deficient on ascorbic acid. You're going to see a patient that has low dopamine beta hydroxylase here, where they have a lot of HVA, but their VMA is kind of on the lower side. And sometimes I'll see this VMA almost like come to like close to zero. And you'll see this HVA to VMA ratio being skewed. There's three different things that can cause this ratio to become skewed. The first is Clostridia overgrowth, because Clostridia produces toxins that can inhibit this enzyme. The second is certain toxins, especially toxins that have phenolic-like structures. And those phenolic structures are like those ring-like structures, such as like dopamine and norepinephrine have, like similar to here. So these are like these are phenolic or benzene rings. Any type of toxins that have these like benzene rings in them can cause this enzyme to be inhibited. The third type of toxin is mutations. There are certain polymorphisms in the dopamine beta hydroxylase gene that can cause a low amount of activity. And so the symptoms for this are like confusion, loss of attention, addiction, constipation, obesity, thyroid disorders. There's a couple different ways you can you can bypass it by producing or by giving a drug called It's kind of a prodrug for norepinephrine. It's called L3-DOPS. The way that I do the most is I provide the cofactors for the dopamine beta hydroxylase, which is mostly vitamin C. For patients that have really low VMA, I usually recommend doing the vitamin C 500 milligrams 2 times per day. And so I had talked about different like ways that you can inhibit dopamine beta hydroxylase, and this is like the first one here, where you have you can see here's the Clostridia. Clostridia produces this toxin called 4-cresol, and you can see this benzene ring here. This will inhibit the dopamine beta hydroxylase, inhibiting dopamine to be converted over to norepinephrine, which then causes too much HVA and not enough VMA to be present. So here is a case study. So you have a patient here with a lot of 4-cresol being produced. And this will result in the patient having a lot of HVA here. So you'll see a patient with a lot of HVA and not a lot of VMA. And really for these patients, the first thing you need to do, besides doing the vitamin C, is treating the Clostridia, which is like there's a lot of different natural products out there that can help clear out Clostridia. So the next thing we're going to talk about is detoxification, which is kind of like my training really was on the interface between genetics and detoxification. So I worked a lot on the enzymes that are responsible for detoxification. And there's probably a lot of you know, there's 2 different routes of detoxification. You got phase 1, which involves your cytochrome P450s, but it also includes a lot of other enzymes, such as one of the other big ones that we're going to talk about is the PON1 enzyme. The other phase is phase 2, which is equally important, because without phase 2, phase 1 is not going to work as well. This is like looking at your like sulfation, glucuronidation, glutathione conjugates. So, and both of these work in concert to like remove toxins. And again, we're back to the methylation pathway. So without proper methylation, you can't have proper detoxification. So you get this MTHFR leading to the production of homocysteine, which then you have cysteine being produced, and then you have glutathione. There's several different types of conjugation reactions here. So you get your phase 1. Phase 1 can lead to 2 different routes. You can either form these like lipophilic molecules here when you add like an oxygen to the your toxin, or you can do a hydrolysis reaction where you add an OH group. This one you then like add glutathione on, and it usually gets urinated out. There's other ways you can add other nucleophiles in, such as sulfation, acetylation, or glucuronidation, and all these cause it to be hydrophilic and then removed via urination. Very frequently, when you talk to somebody that deals with toxicology, you'll hear them talk about xenobiotics. Xenobiotics are any compound that comes from outside the body. A lot of times you'll talk about medications. All medications are referred to as xenobiotics by toxicologists, but other xenobiotics are things that you aren't willingly put in your body, such as like toxicants, such as like pollution and plastics and rubber toxins. And all of these lead to glutathione conjugates. And there's 2 different markers on organic acid test that we measure here, and these are markers 58 and 59. And if a patient is being exposed to a lot of different toxins, you're going to see elevations in these 2 markers here. So you can see here if the pyroglutamic being elevated and the 2-hydroxybutyric being elevated, this patient is using up a lot of glutathione. Patients that I see that used up a lot of glutathione, the most common is mold mycotoxins. I see it in more and more patients every day having elevations in these 2 markers because of being exposed to mold mycotoxins. Other common toxins are like acrylamide, um, different rubber metabolites are very common. And so you can see here, these are a lot of different markers here that are metabolized by glutathione. We're not including mold toxins in this, but we got acrylonitrile, ethylene oxide, xylene, benzene, acrylamide, 1,3-butadiene, propylene oxide, 1-bromopropane. All of these are metabolized by glutathione. This whole chart here is available on our blog at the Great Plains Laboratory. So I recommend everybody go into the resources tab on our website. There is a collection of webinars on our website, as well as a collection of blog posts. One of them is on different toxins. The other one is how to deal with toxins. And you can see here, a lot of the toxins are metabolized by the cytochrome P450, but the other like half are really metabolized by the glutathione. And there's a lot of polymorphisms in glutathione. And right here, I'm just talking about the polymorphisms that deal with the transferase. But I'm really kind of like leaving out the polymorphisms that deal with the production of glutathione. Because even after you're producing cysteine, there's several different polymorphisms that are after cysteine that can cause the production of glutathione to be low. So what are some of the complementary use of the organic acid test for the for different toxins? And some of these are 4 different aspects. You got mold colonization, glutathione status, mitochondrial health, and gut health. And a lot of these can be like showed here on this graph right here. So first talking about gut, colonization. So for mold colonization, Aspergillus produces 2 different types of molecules. One is this 5-hydroxymethyl-2-furoic, which is marker 2 on the organic acid test. And the second one is this furan 2,5-diboxycitric. If a patient has elevations in either of these, it shows that the patient has colonization in either the sinus cavities, or it could show colonization in the gut. Then you really need to like treat with like some type of antifungal. I'd say maybe about 20 to 30% of our patients with elevated mold mycotoxins have elevations in these markers. A lot of patients, however, don't have elevations in these markers. They just absorb the toxins. And the only way you can tell that is by running the urine mold mycotoxin test. On the organic acid test, there's also good markers for looking at environmental toxicity, such as lactic acid, succinic acid. Succinic acid is more for like metals, plastics, mold. Lactic acid is mostly, most of the patients I see elevations in it are patients that are exposed to mold. Lastly, talking about glutathione status here, so patients that are exposed to mold use a lot of glutathione, but then also patients that are exposed to rubber metabolites and car exhaust and acrylamide and metals, they can also have elevations in the usage of pyroglutamic acid. Other aspect of the organic acid test is very useful, especially for patients that have yeast or patients that have oxalate metabolism. And oxalates can cause, crystallize in a lot of different tissues. We see them in the eyes, we see them in the kidneys, we see them in muscles. They can form, here's one forming of staghorn in kidneys. And there's 3 different ways that you can get high oxalates. The first way is yeast. The most common is yeast. The second is genetics. And there's like 3 different genes that are associated with high oxalates. And you got the glycolate oxidase, the alanine glycolic aminotransferase, and the glyoxylate reductase hydropyruvic reductase. these three genes here, if you have a patient that have elevations in oxalates, you might have metabolite mutations in these three. These are kind of less common. I did have two young sisters that came through not too long ago that had oxalates over 1,000. Both of them had oxalates over 1,000, and we figured out that they actually had mutations in the GR-HPR polymorphisms. But the other two ways of getting high oxalates, the most common here is yeast, and the other one is diet. Patients that don't eat enough calcium but eat a lot of foods that are high in oxalates, and we have a list of foods that are very high in oxalates on our website. So if you just go under the organic acid test, there is a list of high oxalate foods under the organic acid test on our website. But more looking at the genes associated with high oxalates are the AGXT, the GR-HPR, and the HOG-A1. This is primary, second type 2, and type 3. So here's a case study here looking at the patient with high yeast. So you got rhabinose here, marker 7. You got about 150 milligrams per gram of creatinine, and then you have a high number 8 here. And you can see here, looking at marker 21, the oxalic, you're going to see very high elevation of oxalates. Most patients that are getting high oxalates from yeast will only show it as marker 21. The few patients I do see with polymorphisms, you will see elevations in most likely 20 and 21. If you see elevations in both of these, that could possibly show that you might need to do some genetic counseling for your patients. Here's the other instigate that case where people can miss it quite frequently. So there's several strains of yeast out there that actually make their own ascorbic acid. So I have a lot of, this gets by a lot of practitioners where on the page 1, you won't see any of the normal yeast markers being elevated, but then you'll see a patient that shows ascorbic acid here, a 1339. This is actually yeast overgrowth. Anything really over 200 is not being caused by supplementation. Vitamin C is metabolized very quickly in the body. It has a half-life of about 3 hours. So if you have a patient that has really high ascorbic acid, especially something like this is being caused by the yeast. Here's a paper from 1972 that's actually talking about how different yeast strains can make ascorbic acid. It says here from 4 yeast have been shown to produce ascorbic acid. So the treatment here for oxalate metabolism, you really want to give high doses of B6, at least 50 milligrams a day. You want to treat the candida if present. You want to do a low oxalate diet. Some of the foods I really see the most common here are nuts and berries, spinach, soy. Those are the ones I really see the most common. Other treatment that's very useful for treating for oxalates, high oxalates, are calcium citrate and magnesium citrate. You want to give these during meals, and this will bind up the oxalates and prevent them from being reabsorbed into the bloodstream. So it's really good talking with all of you today. I hope that you will able to pick up some things. And so if anybody has any questions, I'm sure that people could send me emails. I'm sure that the administrators of the conference can share my email out to people.
Thank you, okay, Matthew. Thank you very much. That was fantastic. Now, I'm sure we've got lots of questions. So I've got one, though, while we get the microphone around. If anyone has a question, put their hand up so we can get the mic. I see B5 elevated a lot when we've got gut stuff going on. Is that the same sort of thing in that certain bacteria can make B5?
It is. It is B5. There are certain bacteria that can make B5, and it's quite common.
Any idea what we're sort of looking at when the B5, the pantothenic acid, if that marker is elevated? What sort of bacterial we might have?
That one I have not been able to like do like a metabolomic study. We haven't been able to like since we only get the urine. I'm not getting the I haven't been able to like culture what the bacteria is, so I haven't been able to identify what strains those are yet.
Okay.
And I haven't found it in the literature yet either.
Okay, thank you. I've looked, but I haven't found anything either.
Yeah.
Yeah. Okay. Do we have a question for Dr. Pratt Hyatt? Yes. Anyone? Yes.
Sorry, it's not specific about the testing, but you mentioned Clostridia treatment, and there's lots of effective natural things that we can use. Can you mention some of them, please?
How about I answer that and say we'll send out the treatment protocol if you're okay with that.
That's fine. I gave it to you, so yeah, that's perfectly all right.
The reason we can't talk about specific products, just ingredients, so Dr. Pratt Hyatt's happy to share that protocol with you, so we'll send it to you tomorrow.
So I'll hope I just give like I won't tell you like what product it is, but the really important thing is doing a pulsing protocol where you like treat every day for 14 days, 3 times a day, and then you do a pulsing where you treat for 1 day and then you take 2 days off. And this is really important so you don't get reoccurrences of the Clostridia, because if you treat it every day, the Clostridia will go into hibernation and hide until after the treatment finishes, and then it'll just come out of hibernation afterwards, and it won't be like susceptible to the treatment if you don't do the pulsing protocol.
Okay, anyone else have a question? Yes.
Hello, thank you very much for your talk. I just wanted to check on the CBS gene, if you don't mind. I've got a gene test in front of me here which has got the C699T variant as a CC, but it's noting that you pay close attention to that, and I noticed that on the one that you were looking at it was TT. So is that an issue with that particular variant? Do you know?
That I can't. I'm sorry. I would have to like look it up through my notes to like know for certain. I can't remember all the different SNPs, so I don't know how that one would be affecting the enzymatic activity or not. Sorry.
Okay. Thanks anyway.
Anyone else have a question? Yes, Kate.
I've got a question about PFAs. We've got a problem with that here in Australia. I know you do in the States as well. Is there anything on the organic acids test that would indicate someone has issues with those?
No, unfortunately not. PFAs are hard to remove from the body. They have a very long half-life. I think they have a half-life of like 20 years, and they don't excrete through urine. So the only way to like test through those is through blood. And right now I'm not familiar with any like commercial tests for those currently.
Do you know what pathway they're detoxified through in the liver?
They detoxify through, they like bind up to the with the bile salts, and you can like remove them through like cholestyramine. Cholestyramine binds to them very well, but there's no like there's no commercial tests to measure them right now.
Okay, thank you.
Any other questions? No. All right, well, please all thank Dr. Pratt Hyatt for his great presentation. Thank you very much.
Thank you.
All right, so we've
come to the end of the weekend. Oh, have a mic, yep. And how are you all feeling? Awesome. Awesome? Fantastic. So before I've just got a quick little summary, but I just want to say a couple of things. First of all, thanks for my team and my girls, stand up, Kate, True, Kristen Soper, Kirsten Wenborn, Sonia Savage, and Yun Si has been helping us out for the world. I've got an amazing team of practitioners. They're absolutely awesome. And they've come on this journey with me, and we've learned a lot together, and it's really exciting. So thank you. Thank you, girls. I want to thank Dr. Penny Caldecott for being our MC this weekend. She's going to come up in a minute and say a few words. But she's been an absolutely fantastic support for me for this conference. She definitely appreciates the fact that, you know, we have to know this stuff. She's really promoted this amongst her doctors. So I really want to thank you for that, Penny. And I definitely want to thank our major sponsors, FX Med and Research Nutritionals. Dennis and Amal partnered me last year in the very first conference, and as you know, it's a really hard thing to get something like this up. So it wouldn't have happened without them. So I want to say thank you. He's a great partner, Dennis, really good. And we've got our other sponsors, our supporting sponsor, Bioceuticals, and all of the exhibitors. I thank you very much. And lastly, I want to thank you for being here. It's great, and I'm excited that you're excited, and that, yes, I know this information is really overwhelming, but you've got to just go and give it a go. You know, you've just got to jump in, and the way you're going to learn is by trying things out. Don't be afraid. Yes, you're going to have reactions, but that's the way that you learn. So this is just a quick 10 slides, that's it, to just sort of try and summarise for you where we're at, because you've heard a lot of different presenters, but in essence, we're all saying the same thing. So the most important thing we have to do is our history, right? We need to talk to the patient in front of us, and we need to find our timeline. I don't know if you've got this in the portal, but we'll put it up tomorrow. So timeline is absolutely key. When did it start? What was going on? What was your mother doing, and where was she when she was pregnant with you? What are you exposed to environmentally? Where do you live? Are there power lines? Is there EMF? All of these questions are really relevant, because without that, none of this makes sense. You have to be exceptionally good at your case taking. And I think this is a really big problem that GPs have. It's the time constraint. You know, when Medicare dictates you have 15 minutes for a consult, you can't do this. And I think that's a really big problem, and it's not of their making. The genetics. So, so we'll start with this. The sort of questions that we want to ask is, did it happen from birth? Because often when someone says, I've had depression my whole life and I really didn't feel great even when I was a young kid, you know that potentially there's some genetic issues going on, or what was going on in utero or in preconception. Any infections, viruses, HPV, EBV, they have to be dealt with. We can't ignore them. So things like transfer factors are going to be one of the first places you'll start to help with that viral load. Exposure to mould. So many of the speakers have spoken to, and that's where the organic acids, I think, is great, because at least you can see markers. However, just because the markers aren't there and someone's got a chronic sinus infection and lives in a mouldy house, then your questioning with your patient is all important. Candida, tinea, thrush. We know now, don't we, the whole weekend, that Candida and fungal infections are probably one of the most important things we need to address, because we're inhibiting certain enzymes like methionine synthase and MAT and DBH that we've got to deal with. We've got to talk about plastic containers, and if they're susceptible through, you can see that susceptibility with CYP1B1 and COMT, then it's going to be crucially important that you say to your patients, you've got to avoid plastics, PCBs. You've got to stop buying food in plastic containers. Don't heat foods in the microwave. Are they eating GMO? And particularly if they've got PON1, you heard Dr. Pratt Hyatt talk about PON1. I think it's one of the most important genes that we can look at for susceptibility. So all the basics that we know, we're still applying, but when we look at the genetic susceptibility, we've got the ability to go down a line of questioning that gives us even more clues, because we can see there's a potential susceptibility. So it's exactly the same thing that Ben Lynch was talking about. Stress, probably one of the biggest utilisers of methyl groups is stress. And we know it downregulates gut function. It downregulates hormones. So, and it's a bit of, I find it's a bit of a catch-22 stress, because I find if methylation improves, the stress, the capability of the patient to deal with the stress is just improved 1,000-fold. But until you improve it, they're still going to be stressed. So we've got to use almost like Band-Aid treatments until we get the methylation under control. But stress is a big one, so is sleep. Cleaning products. What personal care products are they using? They're all very relevant, because our livers are inundated with toxicity at every level. And so from a naturopathic perspective, you know, even 50 years ago, naturopaths, 75 years ago, they're talking about toxins, cleaning up the environment, cleaning up what products you're using, and we've come right back to that now, because it's even more important than we do it now. What's their job? What's their partner's job? Are they exposed to any toxins? Are they printers and dealing with printing material all day? Genetics. So as Dr. Ben Lynch, you know, putting these out and putting them on the planners and circling them is actually a really good strategy. And you saw me in the case studies yesterday, that's exactly what we did. And then you look at what decreases it, what increases it, and what's the cofactor, and is it relevant for that patient? Right? So you can see their susceptibility, but what else might actually be affecting it? If they've got yeast and they've got MTR, is that going to be affecting it? Yes. So we've got to deal with these two and the cofactor elements. As someone asked before, you know, about SSRIs and can we actually do anything to improve them if the patient's on it? Absolutely we can, because it's about ensuring that that underlying biochemistry and the nutrients are at a good level. And usually you'll find that you will get improvement when they're on SSRIs, and eventually they'll be able to come off it because they feel so much better. Some don't, and that's fine. And so when I'm looking at susceptibility, here's my list of genes that I look at for the different pathways. So there's the folate genes that I consider, B12, choline and glutathione. So when you're doing your line of questioning and you see that some of these are coming up for your patients, then you can ask questions around folate metabolism. You can ask questions around B12, neurological, numbness, tingling, brain not working, a lot of things that we can see. Choline, you're going to be asking about memory, you're going to be thinking about myelination of nerves, you're going to be thinking about cellular membrane health. And glutathione, obviously we need to be able to reduce it and get it back from its oxidised form. But if we've got no capacity to really do that, or our susceptibility is there, then we need to be looking at cofactors and what we can do to improve that. And particularly in our environment right now, that's even more prevalent. We've got to do it because we're exposed at every level. Estrogen. So we're considering CYP1B1s and the sulfur and CYP1A1 and COMT. Dopamine, you can see those there, serotonin and energy. So here's your susceptibilities, just plot it out and think about it. We don't want to look at all the genes and consider it all the time. You want to consider the genetics in light of what the patient is coming in with. Does that make sense? Yeah. So if it's not relevant, get on with the next question. Okay, so, and then we need to look at what is inhibiting. And everybody's got something inhibiting. So forget the genes, have a look at the susceptibility, then move on and say, okay, well, is there anything that could be inhibiting? Lipopolysaccharides, candida, heavy metals, environmental toxins. We spent a whole conference last year talking about environmental toxins. So we need to be detectives. We need to be medical detectives and find out what is going on with the patients. And we have got amazing tests to help us work that out. We don't have to work it out on our own. We've got organic acids, we've got stool tests, we've got Marcon's tests, we've got toxicity tests, the GPL tox. We've got so many tests that can help us decide, well, what is going on? What is my next move? And so here's just other things to, gut is key, we know that. But we decide at that first appointment, you know, maybe there's a histamine problem right now because there's too many gut bugs going on. Maybe there's oxalates, maybe there's yeast, is there low hydrochloric acid? Maybe they can't make enough probiotics, maybe there's sulfur sensitivity. So here's what you're going to be also assessing at your first appointment, because there's very few patients, if ever, that don't have some sort of gut problem when they come in. And again, heavy metals, it's a big deal, and so is yeast. So that really, if we could summarise the whole weekend, that's it. So just go and play with it and use it and see how you go. And just work through each patient by taking good case histories and then moving on. So thank you so much for being here. Really appreciate it. And I'd like to ask Penny to come up now.
So thank you, Carolyn, for that great roadmap, practical things that we're going to go away for, and for putting together this amazing conference summit, because I've put together as a convener, I think, 7 conferences, and they're big, hard work to do. And I think this one has been a very successful one, and the biggest success is to be able to go away and be able to start changing what you're doing and how you're thinking about things immediately, which is, I think, what we now have some skills and some knowledge to do. So I like what Ben said this morning, don't get overwhelmed, do one thing at a time. And it reminds me, you know, we've been talking about this kind of matrix and the puzzle and stuff, and it reminds me of a book that won the Booker Prize in 2013, which was The Luminaries. It was written by a young New Zealand woman. Did anyone read that book? In my experience, you had to get about halfway through or more before you understood what the hell was going on. So you just kept reading because it was interesting, but it's like placing the characters and the stories was really difficult, and suddenly it all fell in together. And that's kind of what happens when we're learning new complicated things also in medicine. I think it's we just keep at it and keep learning, and suddenly the picture falls into place. But I think that what we've done over the last few days is certainly helping that picture to fall into place. So I had a couple of things before we just get on to the end of the housekeeping and the thank yous. So as some of you know, I'm the president of the Australasian Integrative Medicine Association, and I know that half the people in the room here are doctors. And it's true that the way we practice and the restrictions and the hypervigilance around what we do is very different from other practitioners, and I think that it's really important to acknowledge and understand that. We recognise that there's a significant amount of stress and fear and reluctance for GPs and other doctors to take on new things or to actually practice differently, because we feel that we're being kind of watched and scrutinised. And we are definitely to some degree. I think that the guidelines that we have that we're meant to be practicing by, are certainly limited in terms of finding out why people are sick and helping them to get better. But the guidelines have been widely criticised even within conventional medicine, and we know that it's been in the front of our medical news and stuff. So guidelines are there to be evolved. Our learning is there to be evolved. I think that what we call integrative medicine now clearly is going to be the medicine of the future. It's evidence and experience-based, and that's what we have to get out there and have to help people understand what it is that we're doing. So in medicine, we do a risk assessment that's evidence-informed for each individual patient. And I think in practice, the risk assessment is for our patients, and it's also for us when we're taking different pathways. And that's very important to do. So the imperative, I think, in being what we might call an integrative doctor now is to be a very, very good conventional doctor. But we also have to have that kind of curiosity that actually drives science. And to be scientific or to be a scientist, you have to have that curiosity and ask the questions, which is what I think we're being compelled to do now with our patients. So in AIMA, just a couple of words, a couple of things. So that the three things maybe that we're doing that are most important now, we're developing integrative medicine pathways for training for doctors. And it's not that we're going to be developing all new educational materials, but we're going to be putting together educational resources that are already together, that we already have, that we will assess as part of a pathway that will also be accompanied by a quite strong supportive mentoring pathway as well. And so when we take that to the RACGP, to other colleges, to the Medical Board and to APRA, we can say, well, this is what an IM endorsed AIMA doctor will look like. This is the type of training that they have done, so that we're actually helping to define what this looks like. Because at the moment, anyone can call themselves an integrative doctor, and we don't really know what that means. So that's really important. We've got an interprofessional working group that has put together letter writing templates for practitioners to write to doctors, because we understand that doctors, even if they're IM doctors, integrative medicine is not about standalone practitioners, it's about how we work together. So these templates are there to train practitioners to write to each other and to write to doctors for the benefit of our patients and also for our own learning. And that project is going to enlarge to help to expose doctors and other practitioners to what kind of skill sets that we all have and how we can work together. And the last thing for doctors is that we're developing an advocacy council. So the advocacy is not so much about getting our message out there, but about how we can practice discerningly, what kind of resources are there to support us in doing that, but also what kind of resources are there if we get into any kind of strife or we get challenged. So the Advocacy Council will go across a number of different organisations, and AIMA will coordinate that. So we're hoping to really make a difference and bring together all the groups in integrative medicine. It's quite a fragmented kind of industry. As you probably know, even amongst naturopaths, there's a lot of fragmentation of different groups and different ways of doing things. But I think that AIMA's job is to really pull together the non-for-profit groups, industry groups, and really work together for the benefit of practitioners, but particularly for this tidal wave of patients who are desperate for a different kind of care that we need to provide in a more comprehensive way. So thank you for listening to that. Become a member of AIMA. We need your support. And we also have some really interesting news coming up for practitioners in terms of practitioner listing on our new website. We have an internationally peer-reviewed journal that's done with Elsevier. John Wardle, who's an academic and clinical naturopath at UTS, is the editor-in-chief. You can put in case studies to the journal. You can put in new ideas that you're thinking about, that you're working with. You can put in models of care as well. So it's not just about research. And so this journal has been coming out 3 times a year. It's just about to go to 4 times a year. You get it as part of membership, but look it up online and be a contributor, because that's the way we're going to get our knowledge and our experience out there as well. So for housekeeping, at the end of the summit, a certificate will be provided to you in recognition of the completed education. And if you go to the registration desk, you can collect your certificate. The feedback forms that we've talked about, don't forget to finish them and to hand them in at the registration desk. And another thank you really for the sponsors who've made this summit possible, and not only possible but such a success. And we'd like to thank our major sponsors, Dennis Schoen from Research Nutritionals and Amal Carl from FX Med, and our supporting sponsor Laura Stewart and the team from Bioceuticals. And for all the exhibitors, Bioceuticals, B Wellness, Cellogic, Grundbiotics, Kingsway Compounding, Nutripath, Nutritional Medicine Limited, Osborne Health Supplies, Research Nutrition, Research Nutritionals and FX Med, Spectrum Ceuticals and Sunstream Infrared Saunas. If we can just give them a hand for being with us and supporting us. There are recordings of the conference sessions available for purchase post-event, and also all the other resources on the website. And we'd particularly like to thank all of our speakers who've just given a whole lot of their knowledge and experience and their own kind of IP to share with us, to use with our patients. So thank you very much for the speakers. And lastly but not least, to thank Carolyn for her incredible effort, not only in organising this event, but the amount of effort she had up here teaching us and guiding us and supporting the other speakers. So thank you, Carolyn. And Lisa Harrison, who was conference organiser, and her team, all of the staff here, particularly the technical team up the back, and for the great food and great environment here. And thank you for all, like Carolyn said, for all being here, and not only be willing and open and curious to learn, but also be dedicated practitioners for your patients. And I'm sure that we'll see you all again at the next summit. Thank you.